Welcome and hello. It has been a while. The last time that we were together in class, we were talking and just finishing up DNA replication and starting to get into protein synthesis. But what I wanted to do was to make a quick video that definitely connects current events of the novel COVID-19 virus to our Unit 7 discussion on gene expression. What I want this video to accomplish is I want for you to really hopefully better understand how viruses fit into life's central dogma, and that even though viruses are debatable, living or non-living, that they absolutely do follow to some extent the central dogma, although certain viruses can follow this a little bit differently than life or living cells will. At the end of this, what I want you to do in Schoology are to answer the two questions. The first question is just for you to kind of summarize what COVID-19 does in terms of how it hijacks your cells, and really make sure that you relate that then to how it goes through the process of entry, and then how the virus will be able to manufacture more viruses, with a highlight on that the gene expression, so protein synthesis for the virus. And then the second question asks you to compare retroviruses to a coronavirus. So they are a little bit different, but pretty similar to one another. But in terms of their life cycle, that life cycle, I want you to be able to analyze and use the two models to compare that are in Schoology as to how they both are going to replicate themselves. That there is a big difference between the two, even though they're both RNA viruses. And then finally, I will link this New York Times article that I'll scroll through with you in Schoology, so you can go back and refer to it. Okay, so how does a coronavirus hijack your cells? So the coronavirus, it's not a new class of viruses, but it has been around for a while. And it wasn't until the early 2000s that scientists really started to take note of the coronaviruses and look for these novel variations of the virus. So you may have heard of SARS or MERS before, but you may have even had a coronavirus before. It does cause a lot of colds, the common colds that we do have, although they are not going to be as severe as the coronavirus, COVID-19 is. Common colds typically then just are going to affect your upper respiratory tract. Whereas COVID-19, there's evidence that it attacks both upper respiratory but also lower respiratory. Those are your lungs, and that's really then what causes the virulence of the COVID-19 variation. Why is it called a coronavirus? Well, it has these spikes on the outside, so it looks kind of like a sun's corona, or a crown, which is where the name comes from. But again, it is important that you note that it is surrounded by a lipid bilayer with these proteins embedded. So therefore, if you use soap and water, that soap will actually pry open this lipid membrane and kill, if we can kill a virus, the virus. Does work much better than hand sanitizer, which does do the same thing about disrupting the lipid bilayer as long as it's 60% alcohol, but doesn't do a great job about getting it off of your hands. So what will happen is this virus is only going to be able to affect certain types of cells, but for the COVID-19, it can affect both upper and lower respiratory cells. It actually will attach onto a protein receptor, the ACE2. This is going to allow for that receptor-mediated endocytosis, and it can enter into your cell. Once inside of your cell, it's going to open up and it's going to release the RNA strand. This viral RNA, though, is fairly large considering other RNA viruses, and it's going to immediately start the translation process. So there's no transcription involved here. It is just going to go right to translation, although there is some replication, but not of DNA, of the viral RNA. Once the ribosomes have manufactured that developing protein, most of that is going to take place on this structure right here. And hopefully, you know, this structure is the endoplasmic reticulum. And it's going to take place on the endoplasmic reticulum so that it can deliver those virus proteins to the next organelle in terms of process of protein synthesis if we want to expel something from the cell. So then it's going to deliver that to the Golgi apparatus. Once it does deliver to the Golgi apparatus, it will then have this membrane that it's going to be shuttled from the ER to the Golgi with, and then these viral proteins, in addition to the newly replicated viral RNA, will be able to go to the surface of the cell and create the new copy of the coronavirus. Then it will bud, and through exocytosis, it will release a lot of these viruses. So this one's really going to primarily go through the lytic infection phase. So it's going to immediately take over and hijack that cell. And then once those viruses are out in your respiratory tract, you can expel them through coughing and sneezing. And that is why it is often encouraged that people that have infections, active infections, are going to wear masks, even though a mask is not going to prevent you from being able to get the virus, since the viruses will be able to pass through most of those masks that one might wear. The immune response, though, typical immune response you would have with any other type of virus, your body is going to recognize it as foreign and start an immune attack. So your macrophage will engulf, and it will then allow for your T helper cells to start an immune response. But one thing that does happen is that your immune system does tend to get overwhelmed, and therefore it overreacts to this virus. And when it does that, it releases a cytokine storm that is going to kill off a lot of cells in the area through inflammation. And then when you have a lot of the fluid and dying cells, it will then make it really hard to breathe. It can lead to pneumonia and then possibly death. When you are thinking about developing some type of virus vaccination, it would follow a very similar protocol. So what you would look into doing is developing these proteins that are found on the virus coat that would allow for your body then to generate its own antibodies, and then would allow for those antibodies to stop this viral infection, primarily by attaching to the surface proteins and preventing that virus from entering in through the ACE2 receptors. But before that happens, again, just make sure that you wash your hands, to keep your distance. Soap is incredibly effective at disrupting the membrane, and these amphipathic soap molecules that look very similar to phospholipids will wedge themselves into this viral membrane, causing it to lyse. So make sure that you stay safe out there, wash your hands, keep your distance, and I hope to see you all very soon.